So yes, I'm Julie and I'm from Maastricht University, and today I will present our work explaining graph neural networks predictions for drug repurposing. So I think we are all aware that there has been a significant attention towards the development, but also utilization of extensive knowledge graphs, and especially for computational drug discovery tasks. And this is mainly due to the reason that knowledge graphs can collect a lot of information from multiple sources. And this allows for storage and also the discovery of complex and hidden relationships between nodes. And this is especially important in the problem of drug repurposing, which aims to find new potential uses for an existing drug. And the problem is often modelized as a link prediction problem. So in order to address the problem of drug repurposing, there have been several methods that have been used, including embedding, rule, and also network-based approaches. But recent developments really promote the use of graph neural networks due to their high capabilities of doing reasoning over expensive knowledge graphs. However, there has been one major challenge or drawback of using graph neural network, and this is the one that graph neural networks introduce interpretability challenges. And this comes from the fact that graph neural network aims to compute new node representations of the nodes, so node embeddings, and it's doing this by aggregating the information from your neighboring nodes. And due to this aggregation strategy, you don't really know which nodes were really relevant in order to get to that prediction. And this is why graph neural networks introduce interpretability challenges. So here I just wanted to show a bit of the workflow of how you train a graph neural network. So you have your input graph, which is the knowledge graph. And then you have your graph neural network, and the output of your graph neural networks are just node embeddings. And since we are doing link prediction for the drug repurposing problem, it suffices to compute, for instance, the dot product between two node embeddings in order to get the prediction. And then you just actually train your graph neural network as any other machine learning model. So you want to optimize your loss function between the predictions and the labels. So here I also wanted to explain a bit more about the GNN layer. So the main idea behind the GNN layer is to compress a set of vectors into a single one. So the idea is really that the nodes exchange information and also influence each other's embeddings. And this is often done in a two-step process, namely message and aggregation. So the idea is that each node will create like a sort of message. So it's a node embedding, and then it sends those node embeddings to its neighboring nodes. The second one is aggregation. So each node will then aggregate the messages from its neighboring nodes. And this is often done by summing the node embeddings up or just taking the average. So to recap, graph neural networks are a powerful tool for machine learning on knowledge graphs. And the main idea behind GNNs are that they combine node feature information with the graph structure by recursively passing neural messages along the edges of the input graph. However, as I already mentioned, there's one main issue with graph neural networks, and that is that the aggregation strategy makes it really difficult to trace the source of those new node representations. So which is why GNNs make it difficult to explain the predictions, and hence GNNs lack explainability. So one related work to ours is the model XPredict. So XPredict has also been presented at this conference last year. And XPredict is a knowledge-based explainable AI approach for drug repurposing. And it focuses on generating cosine similarity-based explanations by calculating the cosine similarity between drug-drug and disease-disease pairs. However, there are actually two major limitations with XPredict. The first one is that it only covers metapaths that are also having drugs and diseases that are very similar to the ones in question. So it may miss potentially relevant but less prominent relationships. The second limitation of XPredict is that it excludes relevant entities like genes and pathways in the generated explanations. So for that reason, we developed a framework for explaining the predictions made by graph neural networks. So our framework takes as input any prediction of a trained graph neural network, and the output is then an explanatory subgraph by taking into consideration drugs, diseases, pathways, and genes. So coming a bit more to the methodology. So here on the right-hand side, we can see the entire pipeline that is used to generate first the GNN predictions and then the node mask. So what we first did, we took the drug repurposing knowledge graph as input, and then we trained 3 different GNN models, such as graph convolutional network, graph attention network, and GraphSAGE. And then we also evaluated the GNNs by computing the precision recall and heats at K. And then we employed Saliency Maps, which is actually an attribution gradient-based method that is capable of attributing an importance cost to the nodes in the network. So after applying Saliency Maps, you know which nodes have been most relevant for a certain prediction. So as data set, we use the drug repurposing knowledge graphs. So on the right-hand side, you can see all the interconnections among entities in the RKG. So we can actually see that there are many different node types, such as gene, compound, disease, pathway, but also molecular function, cellular component, and biological process. And the number between 2 entities actually indicates the number of different edge types. So for example, between compound and gene, there are 34 different edge types. So it's actually a very big knowledge graph. And since we are in the problem of drug repurposing, so a link prediction problem, the aim was to predict the triple compound treats disease. So here's just a small recap of the methodology. So we used graph neural networks to capture the semantics, the graph structure, and also the relationships between the nodes. And then we applied Saliency Map to actually get the importance scores for the nodes in the network so that we know which nodes have been relevant for a certain prediction. And after having those Saliency Maps scores, we use them to create some paths. And then based on the score, we could rank those paths, and then we could add them to the explanatory subgraph. But I will explain a bit more later on that. So Saliency Maps actually originates from computer vision related literature, and it is a pixel attribution method which highlights the most important pixels for a certain image classification. And in our method, we actually want to do the same. So we want to highlight the most important components of the knowledge graph that led to a certain prediction. And the computation behind Saliency Maps is also relatively easy. So it just computes the gradient of the output with respect to the input. So you really want to capture how the output of your model changes as you make small changes to your input. And then Saliency Maps takes just the absolute value of the gradient to capture negative as well as positive inferences. So in order to generate then the explanation, I before mentioned that we predefined some paths that we really wanted to add to our explanatory subgraph. And here are the 3 paths that we are interested in. So the first one was gene participates in pathway, compound binds to gene, and disease associates to gene. So having those saliency map scores, we still needed to come up with an algorithm to actually generate the explanation. So we started off by ranking all the genes based on their saliency map scores in ascending order, so that the first gene is actually the gene that has the highest saliency map scores. And then for each gene, we extracted the 3 paths that we just saw before. And since we know the saliency map scores for all the nodes, we could actually compute the cumulative score of each path and then also rank the paths and and then we just included the top K triples in the explanatory subgraph, and then we continued. Then we just took the second most important gene and we just extracted again the paths, then we ranked the paths and included only the top K paths into the explanatory subgraph. So coming to the experience, so we did the link prediction task. So we wanted to predict compound treats disease. And as metrics, in order to assess the performance of our GNNs, we used precision, recall, and heats at 5 and heats at 10. So precision calculates the ratio of true positives to predicted positives, and recall calculates the ratio of true positives to actual positives. And heats at 5, heats at 10, then for each test instance, we predicted the tail and assessed whether the predicted tail is below 5 or 10. As a dataset, we used the Drug Repurposing Knowledge Graph, and we used as models GraphSAGE, Graph Convolutional Network, and Graph Attention Network. So here we can see the results that were obtained by assessing the graph neural networks. So we can see that good results were achieved using Graph Attention Network, which attained a heats at 5 score of 0.451 and a heats at 10 score of 0.672. And we could also observe that GraphSAGE also achieved very good results by having a high recall rate of 0.992. And high recall is especially important for tasks such as drug repurposing, because in drug repurposing, you really want to minimize falsely predicted links between drugs and diseases. So after assessing the graph neural networks, we still wanted to show the effectiveness of our framework. So we applied the entire framework on a use case for donepezil treats Alzheimer. So we actually took the trained graph neural network and we chose GraphSAGE because it had a high recall rate. And then we applied the algorithm that I showed before to get the explanatory subgraph. So here we can see the explanatory subgraph for donepezil treats Alzheimer. And for those that are not familiar with drug repurposing or Alzheimer, the primary goal of Alzheimer's drugs is to maintain elevated acetylcholine levels, which thereby compensates for the loss of functioning cholinergic brain cells. And figure 2 and also existing literature emphasizes the crucial role of donepezil binding to acetylcholinesterase and butyrylcholinesterase. So here what we can see is that donepezil treats Alzheimer because it binds to acetylcholinesterase, which compensates for the loss of functioning cholinergic brain cells. The second use case that we included was memantine treats Alzheimer. So memantine is commonly prescribed for moderate to severe Alzheimer's disease and is believed to help prevent excess levels of the substance glutamate from damaging the brain. So also, according to our explainability framework and the existing literature, important genes associated with memantine include glutamate receptor and acetylcholinesterase. So there are actually also some limitations of our work. So the first one is that our framework assumes a static and deterministic input graph. The second is that the framework only focuses on local or subgraph level explanations. So you might limit the framework's ability to capture global aspects of the graph. And the third one is that no formal evaluation was conducted for the explanations. So we just showed that the framework works on some use cases, but no formal evaluation was conducted. So as future work, we propose to extend the framework to support maybe various graph types, such as dynamic graphs, to conduct formal evaluation for the explanations, and maybe even user studies with domain experts for better explainability. And to sum up our work, so we developed an explainable AI framework that combines GNNs for link prediction on the DRKG with saliency maps, and we generated explanations that take into consideration drugs, diseases, genes, and pathways. And we showed the effectiveness of applying graph neural networks on drug repurposing. So we showed that good results were achieved using graph attention network. So with a hits at 5 score of 0.451 and hits at 10 score of 0.672. And we also showed the effectiveness of GraphSAGE, which actually attained a high recall rate. And we also showed using GraphSAGE the effective explanations for a case study on Alzheimer's disease. So thank you for listening.
Thank you very much for the presentation, very inspiring. I was wondering, probably a technical question, what's the size of the neighborhood you consider to learn the embedding of the nodes?
You mean when I trained the graph neural network?
Yes.
We always considered the neighboring nodes.
Just one hop?
Yeah, just one hop. Yeah,
okay.
Exactly.
Okay. And another question, since I have the mic. Did you add to filter somehow some of the relationship or type of classes that were coming too frequently?
No, we did not do any pre-processing of the data.
Excellent. Thank you very much.
Other questions for Julie? So I have a question. So you selected GraphSAGE to continue with your experiments because it had the highest recall rate, but it also had the lowest precision rate. Could you say a little bit more about the trade-off between these?
Oh, yes. So you can have, like it's very often the case that you have a high recall and low precision or the other way around. But I thought that in drug repurposing problem, it's especially important to have a high recall because you really want to minimize falsely predicted links between compound and disease.
Okay, so in this case, you can prioritize the recall rate quite safely.
Yes, that's right. Yeah.
Okay, and then I guess this is a bit of a future question work, but you said you haven't done any formal evaluation on the explanations yet.
Yes.
Are you planning to show those graphs to your users, or are you going to show the explanations in some other way?
I think the aim is to really to have some automated way that that says if your explanation is good or not. And we were thinking about maybe comparing our results to DrugMeshDB, which is the database of paths which already shows the mechanism between drugs and disease. And that is something that we might try out later on.